Very good afternoon, students. So, in the last class, we discussed regarding the glucuronic acid pathway. Now, let us move on to the next ED pathway and as well as glyoxylate pathway and few concepts with respect to this Cori cycle and as well as futile cycle. So, first of all, we will focus on this ED pathway that is called as Entner-Diodov pathway. Although this the glycolytic pathway, which is the most common pathway or the route for the conversion of these exoses molecule to the pyruvate molecule, as we have seen in glycolysis, that is glucose getting degraded to pyruvate. So another pathway which is plays a very similar role as that of this particular glycolysis is being discovered is one of this pathway that is called as ED pathway. So here what is going to happen? First of all, this pathway is can be observed in case of the most bacteria and which have this glycolytic pathway and as well as pentose phosphate pathway, but some substitute has been happen with respect to the CD pathway for the glycolysis. The ED pathway, which is generally found in the Pseudomonas, Rhizobium, and as well as Azotobacter, and as well as Agrobacterium, and even few other gram-negative bacteria or the genera also carry out this ED pathway. Very few gram-positive bacteria which also have this pathway, except this Enterococcus faecalis species being exception, which we cannot see in this Enterococcus faecalis species. This occurrence of this Entner-Diodov pathway or ED pathway. So what is going to happen in this pathway especially? So I will tell you the overall aspect of this ED, that is 6-phosphogluconate which is going to act as a precursor here. It is first undergoing dehydration reaction, dehydrated to form 2-keto-3-deoxy-6-phosphogluconate. 2-keto-3-deoxy-6-phosphogluconate can be observed after dehydration, or even it is also called as KDPG. So the key intermediate in this pathway is KDPG, which is further cleaved by this KDPG aldolase enzyme to pyruvate molecule and glycerolate 3-phosphate. So the formed glycerolate 3-phosphate is further converted to pyruvate in the which we can observe in the glycolytic pathway, that is the end product of the glycolytic pathway. So this Entner-Diodov pathway, which is going to degrade glucose molecule to the pyruvate in this way. Once after the formation of glycerolate 3-phosphate, it is further entering into the glycolysis and continues the glycolytic pathway to form pyruvate, or this can also undergo further in the case of the bacteria, other species that is Azotobacter and Agrobacterium species to carry out the fermentation process in order to yield 1 ATP, 1 NADPH and 1 ADH are going to be produced per glucose metabolization. So this how the ED pathway can happen. You can make out in this slide. Glucose molecule further undergoing phosphorylation to form glucose 6-phosphate, 6-phosphogluconate and 2-keto-3-deoxy 6-phosphogluconolactone, right? So you can make out here glucose to glucose 6-phosphate as usual kinase phosphorylation reaction. Further it is undergoing dehydrogenation reaction, and further it is undergoing dehydration reaction and to form 2-keto-3-deoxy 6-phosphogluconate. Then further aldolase enzyme which cleaves this 2-keto-3-deoxy 6-phosphogluconate to pyruvate as well as glycerolate 3-phosphate. Further it is forming this other reaction, other metabolites, right? So that is entering into the glycolysis, where in glycolysis it can continue all the reaction formation of 1,3-bisphosphoglycerate, 3-phospho, 2-phosphoglycerate, phosphoenol pyruvate, pyruvate. And you can see in this right side of this slide, you can make out this pyruvate further undergoes fermentation to form carbon dioxide molecule and acetaldehyde and further ethanol. You can see all these reactions which is going to happen. So you can see in this slide properly the structures of this glucose 6-phosphate, 6-phosphogluconate and 2-keto-3-deoxy phosphogluconate, that is through dehydration, and this is dehydrogenation reaction, and this is aldolase reaction. So to form glycerolate 3-phosphate and pyruvate. So you can make out in this particular reaction what is happening. Water molecule is going to be removed and further it is going to undergo cleavage reaction to form this, and it can these enzymes which in the DD pathway are going to be phosphogluconate dehydrogenase or the KDPG, aldolase which will involve, which are very important enzyme in this ED pathway. So this is all about ED pathway or internal Diodov pathway. Very simple and easy to remember. Very, very important. Please concentrate on this. Now let us move on to this glyoxylate pathway. What is this glyoxylate pathway? Why it is required, and what is glyoxylate cycle? Why it is called so? So the plants and usually the many microorganisms which are going to equip this particular metabolic machinery, which is referred as glyoxylate cycle, which is mainly required in order to convert these fat molecules into the carbohydrates. As such, these plants and other microorganisms cannot undergo other pathways, so that's why they have uses this particular machinery to convert this fat into carbohydrate molecule. What it explains, this glyoxylate cycle, where the anaphylactic reactions which are happen to in order to meet our requirements for the demand of the carbohydrates especially. So this cycle is going to operate for the gluconeogenesis process, that is nothing but synthesis of glucose, that is from fat to glucose molecule or the carbohydrates. This is the modified TCA cycle which can be observed in case of plants and some microbes. This cycle, which is utilizes the fatty acids as the source of the energy in the form of acetyl-CoA molecule, which can be utilized. The acetyl-CoA will be the source for this as a fat or the fatty acids, which will carry out this particular modified form of this TCA cycle, which can be observed in case of plants and some microbes. So what is going to happen in this cycle? The carbon dioxide evolving steps of this TCA cycle were bypassed, and instead of this acetyl-CoA is utilized, which condenses glyoxylate to form malate molecule. So this succinate is going to act as a byproduct for the biosynthesis, particularly which is used for the biosynthesis, especially in the gluconeogenesis, particularly in gluconeogenesis process, that is two molecules of acetyl-CoA is going to undergo dehydrogenation, that is NAD plus, which requires a cofactor to form two water molecules succinate and two coenzyme A, and NAD plus is going to be NAD plus and H plus is going to be formed. So why this glyoxylate cycle should happen? Because the animals including man cannot carry out the net synthesis of carbohydrates from fat. Understanding? So that's why this cycle should happen in, it is going to happen in case of only plants and other microorganisms. So you can see where this cycle is going to happen, or the glyoxylate cycle will happen. What is the location? So this is going to occur in glyoxysomes, especially the cellular with the specialized cellular organelles, where fatty acid oxidation can also be operated over there. So we can make out what are the reactions of this glyoxylate cycle which we can observe. So this glyoxylate cycle, which is an anabolic variant of the citric acid, that's what modified form of this TCA cycle. So acetyl-CoA which are going to be used here, which produced from the fatty acids oxidation especially, further condensed with the oxaloacetate molecule to give the citrate molecule. So what we have observed in case of TCA cycle also, this acetyl-CoA is going to condense with the oxaloacetate to form citrate, right, which is further converted to isocitrate. At this stage, isocitrate is going to be bypassed the citric acid cycle, and further it is cleaved by isocitrate lyase. So where the reaction which is going to happen, that is formation of isocitrate, it is going to bypass there. At this stage it is going to be bypassed, and the citric acid cycle, it is going to bypass the TCA cycle, and further it is, this molecule is cleaved by isocitrate lyase to form succinate and glyoxylate. Further, another molecule of this acetyl-CoA is now going to be utilized to combine with glyoxylate, which is so formed from the cleavage reaction, right, cleavage of isocitrate. So this acetyl-CoA, another molecule, is going to be utilized in order to combine with this glyoxylate to form malate molecule now. So this reaction, which is mainly catalyzed by malate synthesis of malate is happening, right, and this malate so formed, it is entering into the citric acid cycle. So the glyoxylate cycle, which is further resulting in the conversion of these two carbon fragments of acetyl-CoA to 4 carbon compound, that is nothing but succinate, right. So succinate will be the end product. So further the succinate, the malate which is so formed is entering into the citric acid cycle. So this particular 2 carbon fragment, which is further of the particular acetyl-CoA, is going to result in the 4 carbon molecule, that is succinate molecule. So this succinate which is going to be converted, so where in the cleavage reaction you got succinate and glyoxylate, right, isocitrate will be cleaved into succinate and glyoxylate. So this glyoxylate is going to condense with another molecule of acetyl-CoA catalyzed by malate synthase to form malate. So this malate which is so formed entering into the citric acid cycle, whereas this succinate which is formed, right, which is formed, there is a 4 carbon compound, is further converted to oxaloacetate and then further to glucose, which involves the reactions of those, that is gluconeogenesis process. Understanding this oxaloacetate which is going to enter, then further enter into the gluconeogenesis process and it results in the conversion of glucose molecule, that is nothing but the synthesis of glucose which is done from the fat molecule, that is acetyl-CoA. Understanding. So the key enzymes which are required mainly in this glyoxylate pathway includes isocitrate lyase, as we have seen, that is isocitrate which is so formed from citrate is getting cleaved by this isocitrate lyase enzyme, and malate synthase which is required mainly for the synthesis of malate, right, that is by combining with the glyoxylate molecule. Understanding, acetyl-CoA is combining with the glyoxylate resulting in the formation of synthesis of malate plus your malate plus glyoxylate. Understanding. So this malate synthase which is present, these two enzymes are going to be present in the glyoxysomes, especially the plants, right. So this glyoxysomes which mainly imports these fatty acids and as well as this particular aspartate which mainly provides this acetyl-CoA molecule to the shunt. These are acting as a shunt for the importing of this fatty acids and the particular aspartate molecule, right, that is which is formed from the uh, acetyl-CoA molecule. So this aspartate transaminase it is also called as aspartate aminotransferase, which converts further aspartate to the oxaloacetate, permitting this incorporation of this acetyl-CoA into citrate via citrate synthase mechanism, that is citrate synthase enzyme which is from the TCA. So this glyoxylate cycle which goes on simultaneously taking place with the TCA cycle, while this TCA which is going to provide the energy, where glyoxylate cycle provides the succinate for the formation of the new carbohydrate from fat molecule. Understanding, that is succinate which is glyoxylate cycle is providing the more molecules of these two are going to happen simultaneously, but TCA cycle which provides the energy, whereas glyoxylate cycle provides some more succinate molecule for the formation of the new carbohydrate from the fat molecule. So you can make out in this cycle. So this glyoxylate cycle and its relationship with respect to the citric acid cycle, how it is managing the energy reaction which arrows which is indicating representing in the glyoxylate cycle, and the particular blue arrows you can see the citric acid cycle here. Notice that particularly glyoxylate cycle bypasses the two decarboxylation steps of the citric acid cycle, and the two molecules of this acetyl-CoA you can make out here which is entering into the glyoxylate cycle, right. So the glyoxylate cycle, what is happening during each turn, but only one enters the citric acid cycle. The glyoxylate cycle was you can make out one will be acetyl-CoA which is condensing, which is forming condensing with oxaloacetate to form citrate. Citrate will be further converted to isocitrate, that is going to happen. So another molecule of acetyl-CoA is going to condense with your glyoxylate which is going to be formed from isocitrate lyase cleavage, that is isocitrate will be getting cleaved into glyoxylate and which is that glyoxylate and succinate, right. So this you can make out here. So the glyoxylate cycle which is first illustrated by Hans the scientist name Hans Kornberg and Niels Madsen. So this was done this particular pathway which they have illustrated, which was done in the laboratory of this particular Hans Krebs. So they have combined this TCA cycle and as well as modified form of this TCA cycle is nothing but this glyoxylate pathway or the glyoxylate cycle which is going to happen in case of the plants and microorganisms. So you can very clearly you can make out here. So the oxaloacetate formation which is condensing with acetyl-CoA molecule, coenzyme is removed, citrate synthase enzyme, citrate molecule is formed. So this citrate which is acted upon by aconitase enzyme as usual same in case of TCA cycle, what has happened to form isocitrate, then this is further cleaved by isocitrate lyase to form succinate and as well as your glyoxylate. See isocitrate lyase which is cleaving this isocitrate to form succinate and glyoxylate, then this succinate further entering into the TCA cycle. So this further entering into the TCA cycle, where you can make out succinate to fumarate, fumarate to malate, malate to oxaloacetate again there, right? So here you can make out the formed glyoxylate is condensing with one more molecule of acetyl CoA. This reaction is going to happen, hydrolysis is going to happen. So malate synthesis, the enzyme coenzyme A is going to be removed. So coenzyme A is removed, resulting in the formation of malate. So you can make out this malate formation. So this malate further acted upon by this malate dehydrogenase to form oxaloacetate again. It is entering into the gluconeogenesis process, right? So this is going to happen. This is the glyoxylate cycle which can be observed in case of plants and microorganisms. So this is all about glyoxylate cycle and ED. Coming for the next topic, that is Cori cycle. It is also known as lactic acid cycle. So this is metabolic pathway wherein which lactate, which is going to be produced by anaerobic glycolysis, what we have seen, which is going to happen in case of the muscles, which moves to the liver and further getting converted to glucose molecule, which further returns to the muscles and getting metabolized back to the lactate. These are all the things which we have noticed in case of Cori cycle, right?